Good morning. My name is Lorenzo Villani and I'm a PhD student in Sustainable Management of Agricultural Resources, Forestry and Food at the University of Florence. I'm presenting a research I conducted for my master thesis with the title Integrating UAV and Satellite Data to Assess the Effects of Agroforestry on Microclimate in Dodoma region, Tanzania. I will introduce microclimate and agroforestry and the Regreening Dodoma program. Then I will go through the objectives, the study areas and materials, and I will briefly resume the methods used for this research. Then I will continue with the results, and finally I will conclude with the discussion. The main research topic of the research is microclimate management. Microclimate can be defined as the localized conditions of temperature, humidity, and atmosphere in the immediate vicinity of an organism. And it has an important difference from climate, and it is that it can be modified by farmers. This is the reason why some authors argue that modifying microclimate can be an effective strategy to tackle climate change, since it is easier to modify microclimate compared to modifying climate at wider scales. However, this opportunity to increase the microclimatic resilience of the landscapes to tackle climate change is not widely considered yet and is going unattended. Every change made in a landscape and every agricultural practice implemented by a farmer has an impact on microclimate. Agroforestry is known to modify the most important microclimate variables through some processes that are influenced by agroforestry, such as lower wind speed with a windbreak effect, increasing the infiltration and soil moisture retention, reducing the evapotranspiration, reducing the solar radiation, and being a buffer against extreme temperatures. Generally, these processes and variables are studied through field experiments, but in this research we propose a remote sensing approach to study soil temperature in agroforestry systems. The research framework was the Regreening Dodoma program, conducted by the organizations Just Dig It and the Lead Foundation. Its objective is to restore ecosystems, promoting farmer-managed natural regeneration and water harvesting techniques. The farmer-managed natural regeneration is a reforestation technique that takes advantage of the stumps already present on the soil without planting new trees, and this has some advantages. Briefly, the farmers are supposed to select and protect the sprouts coming out from the stumps. Data were collected at the beginning of the project, and therefore the effect of the reforestation were not visible yet. The main objectives of the research were to improve the knowledge of farmer-managed natural regeneration agroforestry systems, to assess the potential impacts of the Regreening Dodoma program, and also to study the land surface temperature tree canopy cover correlation. And finally, we wanted also to propose an operative tree density threshold to be set as objective for the Regreening Dodoma program. 9 study areas belonging to the Regreening Dodoma program spread throughout the whole Dodoma region were selected. For these areas, 9 orthomosaic maps were produced with an unmanned aerial vehicle, a DJI Phantom 4 Pro equipped with a Parrot Sequoia multispectral camera. In addition, one NDVI map was also produced. These maps were representing typical agricultural landscapes of approximately an area of 50 hectares each. The field campaign was done in November 2018, at the end of the dry season, and Landsat 8 and Sentinel-2 scenes were instead downloaded for the dry season of 2018 from April to October. In this slide is shown a brief overview of the methods used for the research. At first, Landsat 8 and then also Sentinel-2 scenes were used to calculate the land surface temperature through the application of the practical single-channel algorithm. Then in a second phase, the orthomosaic maps created with the unmanned aerial vehicle were elaborated to calculate the tree canopy cover. And finally, after the extraction of these variables, a statistical analysis was performed to study the land surface temperature tree canopy cover correlation. The practical single-channel algorithm developed by Wang and others was used to retrieve the land surface temperature. It was selected because it has a good spatial resolution, since it uses Landsat 8, because it is quite easy to be applied, and because it is recent and it has an improved performance compared to previous single-channel algorithms. In this slide are reported the two formula used to retrieve land surface temperature, and the inputs needed are radians from Landsat 8, the emissivity that we calculated through an NDVI threshold method with Landsat 8 data at first and Sentinel-2 data in a second phase, and the atmospheric water vapor that we retrieved from a MODIS product. For the elaboration of the orthomosaic maps, we started producing vectorial grids with the same dimensions and positioning of the Landsat 8 pixels at first, and in a second phase of the Sentinel-2 pixels. On the right are shown the steps and the final result of the elaboration process. The first step was to perform a supervised classification of the orthomosaic maps. Then a visual check was performed to verify the accuracy of the classification and also to exclude cells with houses, roads, rivers, bushlands that we didn't want to include in our analysis. If there were errors in the supervised classification, we proceeded with the manual digitization of the cells containing the errors. And finally, we calculated the tree canopy cover for each cell of the vectorial grid. The calculated data were studied for each individual orthomosaic map without aggregating them. In particular, the land surface temperature tree canopy cover correlation was studied to verify if there was a clear and significant negative correlation. To do this, two analyses were performed. At first, a pixel-based analysis considering the cells individually, and then a class-based analysis in which the cells were grouped in 5 classes of 10% tree canopy cover increments, with a maximum of 50% tree canopy cover. To analyze the data, the statistical analysis was finally performed through linear regression, the Kruskal-Wallis test, and the DAN test. The calculated tree canopy cover averaged 5.1% for the 9 maps analyzed, with a minimum of 0.8% and a maximum of 18.2%. The total number of pixels considered is 4,783. That corresponds more or less to 430 hectares. As can be seen in the figure on the right, the land surface temperatures showed an increasing trend during the dry season, with temperatures much higher in the late dry season compared to the early dry season. We obtained poor results from the pixel-based analysis, while the class-based analysis yielded better results. Considering the linear regression, the two maps with the highest tree canopy cover showed the expected correlation during the late dry season, with values of the R-squared higher than 0.9. The Kruskal-Wallis and the DAN tests were then performed for these two maps, but only the greenest map, the one with the 18.2% tree canopy cover, provided significant results. More in detail, the land surface temperatures of the class with less trees were found to be significantly higher compared to the temperatures of the other classes with more trees. In a second phase, the greenest orthomosaic map was further analyzed with the inclusion of Sentinel-2 scenes to calculate the emissivity and increase the study resolution at 10 meters, and also with an NDVI map that was available for this study area that allowed to produce a more precise classification. In the figure are reported the results of the linear regression and the matrices of significance to the Kruskal-Wallis and Dunn tests that provided significant results considering the late dry season. The average difference in land surface temperatures between the class with less trees and the one with more trees in the late dry season was found to be 1.32 degrees. The expected land surface tree canopy cover correlation was found only in the greenest maps. It is important to point out that most of the maps had cells belonging to all the 5 tree canopy cover classes, but that we found significant results only for the maps with the highest total tree canopy cover, meaning that the total tree canopy cover was important. The poor results can be partially attributed to some confounding factors that have an impact on land surface temperature in addition to tree canopy cover. Among these factors, we can list the soil type and color, the vegetation cover, the micro and mesoclimate characteristics of the surrounding areas, the agricultural practices like tillage or mulching, and the different trees' phenological characteristics. To reduce the impact of these confounding factors, we analyzed a high number of cells, and this might also be the reason why we observed improved results with an increased study resolution and an increased number of cells analyzed for the greenest map. A clear increasing trend in land surface temperature was individuated during the dry season, caused by surface energy balance dynamics and by the partitioning of energy, and also by the different soil cover in the early and in the late dry season, as probably during the early dry season some crops or residues were still covering the soils. To conclude, from our analysis and considerations, we can suggest a threshold of 10% tree canopy cover to achieve an ameliorated microclimate in terms of temperature for the Regreen Dodoma program that correspond to 50 trees per hectare. This was the end of the presentation. I hope you liked it. Thank you for your attention, and for any doubt, I'm leaving my email. And if you want to stay in touch with the Water Harvesting Lab, that is my research group, you can find us on the web using those links. Thank you again.